So I want you to imagine opening a standard text document on your computer. But instead of a report or, a novel, this document contains just a continuous, unbroken string of 4 letters: A, T, C, and G.
Right. Just a massive wall of text.
Exactly. And you press the down arrow to start scrolling. The screen blurs as the letters fly past. And if you were to sit there scrolling through one full page of text every single second, 24 hours a day, it would literally take you an entire lifetime just to reach the end of the file.
Which is just a staggering scale. I mean, it's almost incomprehensible when you really think about it.
It really is, and that is the scale of the human genome. We're talking about roughly 3 billion base pairs. So if you are a biologist trying to find one specific mutation that causes a disease, you are not just looking for a needle in a haystack, you are looking for one specific needle in a haystack the size of a mountain.
Yeah, and it completely defies our normal relationship with reading. Like, we are wired for narrative, right, left to right, top to bottom, one page to the next.
Right, a linear progression.
Exactly. But when you are confronted with 3 billion characters of raw, unformatted biological code, that linear approach just collapses. The data basically becomes a paralyzing wall of noise.
And that is exactly why we are dedicating today's deep dive to you, the listener, who sent in this incredible stack of notes and research. You asked us a fundamental question, which is, how do researchers actually navigate this? Not in theory, but in practice.
Right, the actual day-to-day mechanics of looking at this data.
Yeah. And to answer that, you pointed us toward a specific, highly powerful tool called the Visual Genome browser. So today, we're going to get into the precise mechanics of how this software operates. We're going to explore how computational markers, visual display filters, and sequence alignment algorithms take this impenetrable ocean of letters and transform it into a highly navigable, two-dimensional interactive map.
And that transition from a one-dimensional text string to a two-dimensional visual landscape, that's really the foundational genius of this approach. Because the first hurdle isn't even analyzing the genome.
Oh, right. It's just getting the thing to open.
Exactly. It's simply getting a standard computer to open the file without crashing.
Yeah, I was reading through the technical specs you provided in the notes, and I honestly couldn't wrap my head around the storage requirements. I mean, if I tried to save 3 billion raw characters in a basic text editor, the file size would be massive.
It would eat up all the active memory on a standard laptop.
Right. So how does this software even get the map on the table to begin with?
Well, it relies on an incredibly elegant piece of computer science known as the .2-bit file format. The developers essentially realized that treating DNA like standard English text is wildly inefficient.
Because of the way computers store text.
Yeah. Standard computer text uses a system where every single character, a letter, a number, a punctuation mark, requires a full byte of memory to store. And a standard byte consists of 8 individual bits.
And those are the fundamental zeros and ones of computer code, right?
You got it.
So 8 slots of zeros and ones just to tell the computer, hey, this is the letter A.
Correct. Which makes perfect sense if you need to represent the entire alphabet, uppercase and lowercase, plus numbers and symbols. But DNA only has 4 letters: adenine, cytosine, guanine, and thymine.
Right, just the 4.
So you don't need 8 bits to represent 4 options. You really only need 2. Because in binary code, 2 bits give you exactly 4 unique combinations: 0, 0, 0, 1, 1, 0, and 1, 1.
Oh, wow. I see. So you can map 0, 0 to adenine, 0, 1 to cytosine, and so on.
It is a perfect one-to-one translation. So by assigning each DNA base to a 2-bit binary state, you can basically cram 4 distinct DNA letters into a single standard 8-bit byte of memory.
That is a massive compression ratio.
It really is. It shrinks the entirety of the human genome down to about 796 megabytes.
Which is what, smaller than a standard high-definition movie file?
Easily.
That's wild. You can hold the code of human life in the active RAM of a cheap tablet. But compressing the data just makes it manageable for the computer. It doesn't really make it readable for the human, does it?
No, not at all. And that's where the shift from text to a 2D map comes in. The software doesn't just display the compressed text. It draws a picture based on something called GC content.
GC content. Okay, this is where we bridge computer science and structural biology, right?
Exactly. So the software analyzes small rolling blocks of the DNA sequence, say chunks of 20 to 50 bases at a time. And within each block, it calculates the ratio of guanine and cytosine bases compared to adenine and thymine bases.
Okay, but why does it care specifically about G and C? What makes those 2 letters so special?
It comes down to the physical chemistry of the double helix. The two strands of DNA are held together by hydrogen bonds between the complementary letters. Adenine always pairs with thymine, and they connect using two hydrogen bonds.
Okay, A and T have 2 bonds.
Right. But guanine and cytosine connect using three hydrogen bonds.
Oh, I see. So a GC pair is physically clamped together tighter than an AT pair.
Exactly. It creates a region of higher structural density. The physical melting point of the DNA is actually higher in regions packed with G and C.
That's fascinating.
Yeah. So the Visual Genome Browser takes that hidden chemical density and translates it into visual information. It calculates the GC average of a block of code and renders a pixel on your screen. And the higher the GC content, the brighter the green intensity of that specific pixel.
The moment I understood this from your notes,
Mhm.
my entire perspective shifted.
Mhm.
Because you aren't just looking at letters anymore,
Yeah.
You are instantly perceiving the structural topography of the chromosome.
It completely changes how you interact with it.
Yeah, because you can zoom out and literally see these glowing, dense green bands where the physical DNA is tightly bound and they're separated by dark voids where it's mostly looser A and T pairs.
Right, so you have established the terrain. The one-dimensional string has basically been wrapped into a two-dimensional tapestry of structural density. But of course, a map is only useful if you possess the tools to navigate it.
Exactly. Let's explore those tools. I want to go back to an analogy. Imagine you were trying to find a tiny, independent, unlisted coffee shop in a sprawling, chaotic city. You don't have street signs. You don't have GPS coordinates. All you have is a printed list of 1 billion street addresses in a row.
You would wander forever. It'd be impossible.
Right. To find anything, you need a way to drop a pin. You need to mark where you are and where you've been. And in this software, that is handled by the marker system.
Yeah, the interface provides a highly specific set of navigational anchors. Users have access to radio buttons, simply labeled 1, 2, and 3, that control visual markers.
And the notes detail that marker 1 drops a blue circle, marker 2 is a green circle, and marker 3 is a red circle. I assume you just double-click on the 2D green map and the software plants the flag.
That is a mechanical action, yeah. But what is critical here is the visual design. When you are zoomed out, looking at millions of base pairs represented by pixels, data density is your enemy.
Because it's too cluttered.
Right. If you drop a marker, you don't want a massive icon obscuring the very biological feature you are trying to study.
So what happens if I need to mark 2 things that are incredibly close together? Say I drop the red circle, and then I need to drop the green circle on the exact same base pair. Does it just overwrite the red one?
The software actually anticipates that. The markers are designed to nest concentrically.
Like a target.
Exactly like a target. If you place the green marker on the exact same coordinate as the red marker, the green circle simply sizes itself to fit perfectly inside the red one, creating a bullseye effect.
Oh, that's really smart.
Yeah, so your view of the underlying sequence remains entirely unobstructed, regardless of how many navigational pins you drop in one location.
I love that attention to detail. But there's another marker mentioned in the source material that seems completely different from the blue, green, and red ones, the P marker. It sounds like it isn't just a spatial bookmark.
It isn't. The P marker represents a shift from spatial navigation to active biological querying. When you select the P marker and drop it on a specific location, you are asking the software a fundamental question about the sequence at that exact coordinate. You are checking for an open reading frame, or an ORF.
Okay, can we break that down? What exactly is an open reading frame in this context?
Well, the cellular machinery that reads DNA and builds proteins, the ribosomes, they don't just start building randomly. They look for specific start signals, kind of like a capital letter at the beginning of a sentence. And they stop building when they hit a stop signal, like a period.
Makes sense.
So an open reading frame is a stretch of DNA that has a valid start signal, a valid stop signal, and a continuous sequence in between that isn't interrupted by random premature stop signals.
So it's a stretch of code that could theoretically be translated into a functional protein.
Exactly, and the P marker tests for that instantly. Instead of having to cross-reference an external database or, load a heavy annotation file, you just drop the pin. The software immediately analyzes the raw code starting at that exact base pair and tells you if a valid protein could physically exist there.
It is a rapid-fire test for biological potential.
Right. It's incredibly efficient.
Okay, I have a logistical question about all of this. Dropping a pin on a digital map, like a standard street map application, makes sense to me because the map is a static image. It is fundamentally a 2D picture with an X and Y axis. But we established that the genome is a continuous, one-dimensional string of 3 billion letters.
Yes, a very long string.
So if I drop a blue marker on base number 1 million,
Yeah.
and then I scroll my screen downwards to base number 2 million, well, sorry, let me rephrase. How on earth does the software know where I am vertically versus horizontally? How do you map a 1D string onto a 2D grid and keep the math straight?
The software manages this by wrapping the sequence, much like the scan lines on an old, bulky cathode ray tube television.
Oh, or like reading a paragraph in a book. You read left to right, hit the margin, and your eyes snap back to the left side of the next line down.
That is the visual representation, yes. But under the hood, the software never loses track of the linear reality. Every single pixel on that screen corresponds to a mathematically exact index number in that 3 billion letter string.
So it's always just a number to the computer.
Exactly. When you double-click to place a blue marker, you aren't really dropping a shape on a picture. You are anchoring a specific numerical index in the software's memory.
So if I click on base 1 million, the software just remembers the number 1,000?
Yes. And then, as you physically move your mouse cursor away from that blue circle, the interface provides dynamic spatial awareness. A small numeric display appears next to your pointer, and the software continuously calculates the mathematical difference between your anchored index and the index of whatever base your mouse is currently hovering over.
So as I drag the mouse down the screen, it's doing the math in real time. It calculates the X difference, how many bases left or right I am from the pin, and the Y difference, how many wrapped lines up or down I have traveled.
It basically converts a linear abstraction into measurable spatial geography. You can visually explore a dense region of the chromosome, glance at your cursor, and know that you are exactly 4,522 bases away from your original starting point.
And what if I scroll completely off the screen, like if I wander so far down the DNA strand that my blue pin is totally gone?
You simply use the jump function. With a single click, the software instantly snaps the high-resolution, zoomed-in DNA view straight back to your anchored marker.
Oh, that's handy.
Yeah, you can use these markers as spatial wormholes, basically, allowing you to traverse massive expanses of the chromosome without ever losing your geographic context.
That spatial awareness is critical, but it leads to an obvious problem, I think. Marking a spot and measuring the distance to another spot is incredibly useful, provided you know what you are looking for.
Right.
But if you just open the human genome and start scrolling blindly through millions of glowing green pixels, hoping to stumble upon a relevant gene, your eyes are going to glaze over.
Absolutely. The signal-to-noise ratio in a raw genome is paralyzing. I mean, the vast majority of the human genome does not code for proteins, so to find meaning, you must possess the ability to filter out the noise.
Which brings us to a massive upgrade in our navigational toolset: advanced display filters. If the markers are our dropped pins, the display filters are how we control what actually shows up on the map.
Precisely. The interface features a filter input field located in the top right corner, and it appears deceptively simple, like a standard search bar. But its function is radically different. A normal search bar highlights text, but this filter field fundamentally alters the 2D visual display of the entire application.
I want to illustrate this with a scenario based on the sources you provided. Let's say we have our green topographical map, and we ask the software to load an overlay of every known annotated gene. There are tens of thousands of them.
It would be a mess.
Exactly. The screen would be a chaotic mess of overlapping labels and colored blocks. It would look like a crowded highway at rush hour.
Visually overwhelming, yeah.
But then you navigate to that filter input field and you type in a specific name. The notes use the example of PLR2A.
Which is the symbol for RNA polymerase II. It's the incredibly important enzyme responsible for transcribing DNA into messenger RNA.
So the instant you type PLR2A and press enter, everything changes. The software visually obliterates every single other gene annotation on the entire chromosome. All the visual clutter just vanishes, illuminating only the specific gene or genes that match your input. You are left with a clean map and a single glowing destination.
It's amazing. It isolates your target within its macrospatial context, but, the true power of the system emerges when you move beyond simple name searches and begin utilizing operators.
You mean like the exclusion tools, the minus operator?
Exactly. Consider the physical structure of the DNA double helix. It consists of two complementary strands running in opposite directions, a positive strand and a negative strand.
Right, they run parallel but opposite.
Yeah, and genes can reside on and be read from either of these strands. So if a researcher is solely focused on the positive strand, viewing the negative strand data is simply a distraction.
So they type minus strand minus into the filter field.
Instantly, every gene located on the negative strand is wiped from the visual display. The software parses the command, identifies the property associated with every annotated element, and just hides anything that matches the exclusion criteria.
That is so clean. I was struck by another filter mentioned in your notes, too. Protein plus R-median. If I'm understanding this correctly, a lot of things we call genes don't actually build proteins, right?
That is a crucial distinction, yes. The genome is literally littered with features that are transcribed into RNA but never actually translated into a physical protein. You have
Right.
transfer RNAs, ribosomal RNAs, and a massive graveyard of what we call pseudogenes.
Pseudogenes, like fake genes.
Basically, yeah. They are essentially genomic fossils. They are stretches of DNA that look incredibly similar to functioning genes. They have the right sequence structure, but they have accumulated mutations over evolutionary time that render them broken.
Broken how? Like, they don't work anymore.
Great. They lack a valid start signal, or they have premature stop signals. They are just dead code.
So if I am a researcher trying to figure out how a cell builds its physical structures, I really don't want to be looking at dead code or structural RNA. I only want to see the blueprints for actual proteins.
And that's where the filter comes in. By typing protein plus into the filter, you instruct the software to evaluate every single annotation. It checks for that open reading frame we discussed earlier, and if the gene does not possess the capacity to code for a physical protein, it is removed from the screen.
So it just strips it all away.
Yes. You filter out the pseudogenes, the RNA genes, the regulatory elements, leaving only the protein coding architecture.
The analogy that came to mind while reviewing your notes is a pair of high-end noise-canceling headphones. Imagine you are sitting in the middle of a chaotic symphony orchestra. The brass is blasting, the percussion is crashing, the woodwinds are screeching. It's
A
deafening,
wall of sound.
right? But you only want to hear the string section.
Mm-hmm.
So you type minus strand minus to mute the brass. You type protein plus to mute the percussion. And suddenly, in the middle of all that chaos, you are only listening to the violins.
That is a perfect analogy. And that isolation allows you to perceive patterns that were previously hidden in the noise. When you strip away the non-coding clutter, you stop looking at individual letters and start seeing the macro structures of the genome.
Like the neighborhoods?
Exactly. You can visually identify high-density gene clusters, neighborhoods on the chromosome that are absolutely packed with protein-building instructions.
And on the flip side, you can see the empty spaces, right? The genetic deserts.
The centromeres are a prime example of that. These are the constricted regions of the chromosomes, the pinched wastes that play a mechanical role during cell division.
A little X shape in the middle.
Right. When you apply standard gene display filters, the centromeres manifest as massive dark voids on your map. They are largely devoid of annotated protein coding genes because their function is structural, not instructional.
That makes total sense. Now, this makes me wonder about the limits of these overlays. The notes mention that you aren't restricted to standard gene annotations. You could overlay epigenetic data or gene expression data.
Yeah, the software architecture allows for really dynamic data integration. Epigenetics refers to the chemical tags like methylation that the cell attaches to the DNA to turn genes on or off without changing the underlying letters.
It's like highlighting or redacting a text document.
Exactly. And gene expression data tells you how active a gene actually is in a specific tissue. The notes mention the Human Protein Atlas as a source for this.
So let's run with a hypothetical scenario based on that. Imagine you are researching lung cancer. You could load up the normal, healthy lung tissue data, and the software colors the genes on your map based on their standard baseline activity.
Right.
But then you use a threshold slider within the filter tools. You basically tell the software to hide all the genes that are behaving normally. You only allow it to light up the genes that are severely dysregulated, the ones that are cranked into overdrive or completely shut down in a tumor cell.
And what you have achieved there is profound. You are no longer viewing a static, generic map of human DNA. By applying that specific biological filter, you have transformed the entire chromosome into a visual heat map of a disease state.
Wow.
You can visually isolate the exact genetic neighborhoods that have gone rogue, providing immediate spatial context for the pathology.
It's an incredible way to synthesize data. But I do see a limitation here. Everything we just discussed relies entirely on the scientific community, having already discovered the gene, mapped it, and given it a name.
Yes, it relies on prior knowledge.
It relies on the annotation files being complete. What happens if you are off the edge of the map? What if you are hunting for raw sequences of DNA that haven't been categorized yet?
Well, when you move into unannotated territory, display filters become useless. You can't filter for a name that doesn't exist. You have to transition from visual filtering to raw motif searching. You literally have to hunt the code itself.
Okay, this brings us to the main search box, which is activated by the classic shortcut, CTRL-F. But based on your notes, this is a lot more sophisticated than the find function in a web browser.
Oh, absolutely. The challenge of searching raw biological code is that the input data can be incredibly messy. Researchers often copy sequences directly from PDF or academic papers.
And those usually format weirdly when you copy-paste.
Right, the text might be riddled with spaces, line breaks, or structural numbers. So the main search box provides a suite of automatic formatting tools to handle this. You can click F to instantly strip out all non-biological characters from your pasted text.
That's super convenient.
You can use N to force the software to interpret the text as nucleotides, or P to force it to interpret the text as amino acids for protein searching.
And I noticed there is an auto-detect function. If I just paste a sequence like ATCG, the software knows it's DNA. But if I paste a sequence that starts with an M-methionine, the amino acid that acts as the start signal for proteins, the software intuitively recognizes the amino acid alphabet and dynamically adjusts its search parameters.
It's smart enough to know what language you are speaking, basically.
Yeah, but the tool that really stood out in the notes as absolutely essential is the RC button, the reverse complement tool.
That tool is huge. It addresses a fundamental reality of biology that computer science often struggles with. DNA is a double helix, and its two strands are anti-parallel.
Right.
Think of a divided highway. The traffic on one side flows north, and the traffic on the other side flows south.
In biology, they call that the 5' to 3' direction. Right.
Exactly. So when a researcher searches for a specific sequence, say a binding site where a transcription factor attaches to the DNA, that sequence might exist on the top strand, reading left to right, but it is equally likely to exist on the bottom strand, reading right to left.
So if I just paste my sequence and hit search, I'm only looking at northbound traffic. I'm basically missing half the potential matches.
And this is where the RC button is critical. When you paste your sequence and activate the reverse complement tool, the software performs two distinct operations simultaneously. First, it generates the chemical complement. Every adenine becomes a thymine, every cytosine becomes a guanine.
It swaps the partners.
Yes. And second, it physically reverses the entire sequence, reading it backward. It then simultaneously searches the genome for your original sequence and its reverse complement.
That guarantees you're checking both sides of the highway. But even with that, hunting for raw code seems wildly complex because, well, biology isn't a rigid computer program. Computer code has to be syntactically perfect or it crashes. Biological code is fluid. It mutates constantly over generations.
It's very messy.
So how do you search for a pattern when you know the pattern is probably misspelled?
You utilize motif searching with wildcards and bracket notation. You define the acceptable parameters of biological fuzziness.
Let's break down how that actually looks in the interface. Let's start with a basic wildcard.
Okay, the simplest wildcard is a period or a dot. If you are searching for a binding motif, but you know that the exact base at the third position doesn't actually matter for the protein to bind, you simply insert a dot at that position in your search string.
So I just type AC.G.
Right. You are telling the search engine, I need an A here, a C here, but at position 3, I will accept absolutely any nucleotide, A, T, C, or G. Just make sure the rest of the sequence matches.
That offers flexibility, but it's quite broad. What if you need to be specific about the mutation? The notes mention bracket notation.
Bracket notation allows for precise chemical definitions. If a generic dot wildcard is too broad, you define a specific set of acceptable bases. By typing open bracket C, T, A, close bracket into your sequence, you issue a highly specific command.
So I type the brackets with those letters inside.
Yes. You tell the software, at this exact coordinate, the nucleotide must be a cytosine, a thymine, or an adenine. If it is any of those three, highlight it. But under no circumstances will you accept a guanine.
Wow. You are computationally defining the exact limits of chemical flexibility allowed at a specific biological binding site.
It gives you incredible control.
It does, but the feature that truly blew my mind in the notes is the mismatches function. It basically takes this concept of biological flexibility and amplifies it. You can literally paste a massive sequence and tell the software, find this pattern, but I'm giving you permission to make a specific number of errors.
The mismatch allowance is basically acknowledging the reality of evolutionary time. Binding motifs or ancient genomic structures aren't uniformly identical across the 3 billion bases. They possess a consensus sequence, like an ideal average version, but in reality they drift and degrade.
Over time, yeah.
The mismatch feature allows you to find the distant cousins of your sequence, not just the identical twins.
The sources provided a beautiful example of this involving telomeres. Telomeres are essentially the protective plastic caps at the ends of your shoelaces, but for your chromosomes. They stop the delicate ends of the DNA molecule from fraying or fusing with other chromosomes.
And they achieve this structural protection through sheer repetitive bulk. The telomere is composed of a massive repetition of a simple 6-base sequence.
TAACCC, over and over again. So in the example provided, you navigate to the 5' end, the very beginning of a chromosome. You type TACCC into the main search box. Immediately, the visual map lights up with red highlights showing thousands of perfect matches capping the end of the structure.
Which is exactly what you'd expect.
But then, you use your navigational tools to fly all the way across the entire chromosome to the 3' end, the absolute opposite tail. You hit that RC button we talked about. The software reverse complements your search into GGGTTA.
And the opposite end of the chromosome illuminates.
But the interface design here is what's brilliant. It highlights these reverse complement matches in bright orange, visually separating them from the forward matches in red. You can zoom out and physically see the structural caps holding the chromosome together on both ends, color-coded by their strand orientation.
It is a stunning visual confirmation of the underlying biochemistry. But, the telomere search is relatively clean because the sequence is short and highly conserved. The true test of the mismatch system is demonstrated in your notes with the ALU element search.
Right, ALU elements. So for context, ALU elements are basically parasitic pieces of DNA. They are jumping genes, basically retrotransposons, that have essentially been copying and pasting themselves all over our genome for millions of years of primate evolution.
Yeah, they make up roughly 10% of the human genome. It's crazy.
In the example provided, a user takes a known consensus ALU sequence that is a massive 288 bases long. They paste this entire block of code into the search box while analyzing chromosome 1. But here is the critical step. They set the mismatch allowance as 60.
They are telling the computer, find this 288-letter paragraph, but I will allow you to get up to 60 of the letters completely wrong.
Now consider the computational burden of that request. When you search for an exact match, the software uses a pre-built index. It's like looking up a word in the glossary of a book. It's instantaneous.
Right.
But when you allow 60 errors in a 288-character string, an index is useless. The software has to dynamically scan the raw, unindexed code of the entire chromosome.
And that takes a ton of processing power.
I had to look this up when I was reading your notes, because I didn't understand how the computer wouldn't just freeze. The notes mentioned it utilizes a brute force Boyer-Moore search algorithm. How does that prevent the system from crashing under the weight of those mismatches?
The brilliance of the Boyer-Moore algorithm lies in its ability to skip characters. If you are using a standard search to find the word AEPLE in a text document, the computer checks the first letter. If it's an A, it checks the second letter.
Sequentially.
Exactly. And if you are searching billions of letters, checking every single position sequentially takes forever. But the Boyer-Moore algorithm compares the search string from right to left.
Wait, backward.
Yes. It aligns your search term with a block of text and checks the last character first. If you are searching for APLE, it checks the text against the E. If the letter in the text is a Z, the algorithm knows immediately that the word APLE cannot possibly be there.
Oh, wow.
And furthermore, because Z does not appear anywhere in the word APLE, the algorithm knows it can safely skip the entire 5-letter block and jump ahead. It doesn't waste time checking letters 1 through 4.
Oh, that is incredibly clever. By finding mismatches early, it leaps forward in massive chunks instead of inching along base by base.
Precisely. By implementing Boyer-Moore, the Visual Genome browser can dynamically scan hundreds of millions of bases, checking every possible combination that fits within that chaotic 60-error margin, in a fraction of the time a standard linear search would require.
And the result of that massive computation, the software finds 6,062 occurrences of that mutated ALU element scattered entirely across chromosome 1. The visual map is completely littered with them. You were basically staring at the evolutionary scars of a jumping gene, multiplying and degrading over millennia.
It's profound to look at.
But this brings up a massive point of skepticism for me. If I can throw brackets and wildcards into my sequence, and I can tell the software, hey, allow 60 errors in this paragraph, aren't I just engineering a search that is so impossibly broad that I am guaranteed to get false positives?
That's a valid concern.
If you allow enough typos, eventually random letters look like a word. How do you distinguish a true biological structure, like a degraded ALU element, from pure statistical noise? A random string of A's, T's, C's, and G's that just happen to match your sloppy criteria by pure chance.
That is the existential dread of bioinformatics, honestly. Discerning signal from noise. But the developers of this browser anticipated this exact problem. When you execute a search with a high mismatch allowance, the software doesn't just return the locations. It calculates and prominently displays the mathematical probability of finding that specific pattern with that exact number of errors completely by random chance.
Oh, so it does the statistical math for you in the background.
It is a vital safeguard. Because if the software tells you that the probability of a random match is 1 in 10, then those 6,000 ALU hits on your screen are almost certainly garbage. They are statistical mirages.
Right.
But if the calculated probability of a random match is 1 in a billion and you still find 6,000 occurrences, you can be highly confident that you have uncovered a true biological phenomenon. Allowing mismatches is a calculated mathematical risk, but it is deeply necessary.
Because the code is messy.
Exactly. The code of life is not a pristine binary file. It breathes, it breaks, it mutates. You have to design tools that embrace the messiness to discover the truth.
Okay, so we have covered marking our territory, filtering the visuals, and hunting down raw, messy sequences. But finding a raw sequence is only one part of the puzzle. What about navigating the complex internal architecture of the genes we already know?
That requires a transition from raw motif searching to predictive structural navigation. We need to examine the tools designed for the fast track.
Let's draw a sharp contrast here based on the notes. The raw pattern search we just discussed using CWL-LRF is a brute force algorithmic hunt through the actual letters of the sequence. But the gene search box, which is activated by C2OLG, operates on an entirely different level. It doesn't scan the letters. It queries the annotated index.
Yeah, it functions very similarly to the predictive text algorithms in modern web search engines.
Right, like Google autocomplete. As you start typing a common gene name into the CTOL-G box, let's use our earlier example, PLO2A. The moment you hit the letter P, a drop-down menu dynamically populates. You type POL and the list instantly narrows, querying the database of thousands of known aliases and official gene symbols in real time.
And the execution is instantaneous. When you identify your target gene in that drop-down menu and press Enter, the software executes a dual action snap. It instantly teleports both your macro-level 2D GC content map and your micro-level high-resolution DNA sequence view directly to the physical coordinates of that gene on the chromosome.
You are instantly transported to the correct address in the city.
Yes.
But arriving at the address is just the beginning of the journey. Human genes are massive, bafflingly complex structures. They aren't just solid, contiguous blocks of coding instructions. They are fractured. You have exons, which are the actual crucial segments of DNA that code for the protein. And these exons are separated by vast, sprawling oceans of non-coding DNA called introns.
Right. The introns are transcribed into RNA, but they are subsequently spliced out and discarded before the final protein is constructed.
So if you want to understand how a gene actually builds a protein, you have to focus on the exons. But if you just try to use your mouse or scroll wheel to pan through a gene, you are going to spend 99% of your time endlessly scrolling through thousands of bases of intronic junk DNA just to find the next tiny coding segment.
It's incredibly inefficient.
So what's the solution?
This is where the keyboard navigation mechanics introduced in the Visual Genome browser become truly revolutionary. The software deeply integrates structural navigation directly into the physical keyboard. For simple panning, if a researcher needs to move quickly across a region, holding down the Shift key while tapping the arrow keys increases the scrolling speed across the genome by a factor of 10.
It's like shifting the map into high gear.
Exactly. But the true innovation occurs when you need to navigate the internal architecture of the gene itself. The software maps structural jumping to the less than and greater than keys on your keyboard.
The little arrows on the comma and period keys.
Right. By pressing the greater than key, you instantly jump from the start of your current exon directly to the start of the next exon in the sequence. You completely bypass the thousands of bases of non-coding introns that separate them.
I was thinking about how to visualize this. And the best analogy I could come up with is watching a movie on a DVD or a streaming service. The movie itself, the plot, the characters, the story that represents the exons. But before the movie, and sometimes in the middle, you have unskippable FBI warnings, studio logos, menus, or blank black screens. Those are the introns.
That captures the functional dynamic perfectly.
When you hit the next chapter button on your remote control, you don't fast forward to the blank spaces at 2x speed. You instantly teleport to the exact frame where the next scene of the actual movie begins. That is exactly what the greater than key does in this software. You are chapter skipping through the biology of the gene. You are bypassing the commercial breaks to get straight back to the narrative code.
And to understand the profound impact of this feature on a bioinformatician's workflow, we should look at the example provided in the source material regarding the gene TTN. TTN codes for a protein called titin. Titin is the absolute largest protein produced in the human body. It basically acts as a giant molecular spring that gives our muscle tissues their elasticity.
And fittingly, the blueprint for the largest protein is an absolute monster of a gene.
It is structurally staggering. The TTN gene spans roughly 281,000 individual bases of DNA, but that sequence is severely fractured. It is broken apart into 312 separate, distinct exons.
312 chapters in the movie.
Yes. If a researcher wants to examine the coding sequence of titin, attempting to manually scroll through 281,000 bases to find those tiny coding islands is an exercise in futility. It would take hours of tedious screen navigation.
It's mindless scrolling.
Right. Instead, they utilize the fast-track workflow. They press Ctrl-L-G, type TTN, and instantly snap to the gene's coordinates. They right-click on the visual representation of the gene and select Jump to First Exon.
And from that moment on, they just sit back, rest their finger on the keyboard, and tap the greater than key. Tap, tap, tap.
With every single tap, the high-resolution DNA view snaps to the precise boundary of the next coding segment. But this is not merely a tool for speed, it is a tool for deep biological context. Because every time you land on a new exon, you are staring at the exact physical boundary where the intronic junk ends and the coding exon begins.
The splice sites.
Precisely. You can visually observe the physical chemical signals embedded in the DNA code that dictate where cuts should be made. Usually, an intron begins with the letters GT and ends with the letters AG. By chapter skipping with the keyboard, the researcher is no longer just reading data, they are actively simulating the physical action of the cell's spliceosome. That's the massive molecular machine that physically cuts the RNA transcript at those signals and pastes the exons together.
And this gets even more complex, right? Because the cell doesn't always cut and paste the exons in the exact same order every time. We have alternative splicing.
That is a critical layer of complexity. A single gene sequence does not always equal a single protein. Depending on the tissue type or the developmental stage of the cell, the spliceosome might choose to include exon 4 but completely skip exon 5.
Wow.
This allows one single gene to produce multiple different structural variations of a protein. Titin, for example, is spliced differently in cardiac muscle compared to skeletal muscle.
And the Visual Genome browser actually accounts for this. The notes indicate that if you are parked on a specific location where multiple different splice variants exist, you can use the plus and minus keys on your keyboard. Pressing them instantly cycles your view through all the different overlapping transcripts, all the different alternative splicing patterns that are documented for that exact coordinate.
It fundamentally changes the user's relationship with the underlying data. You transition from a passive observer of a static text document to an active participant. You are essentially playing the DNA structure like a musical instrument, using the keyboard to rapidly simulate the complex, dynamic cellular machinery that processes the code.
It turns data analysis into an incredibly tactile, interactive experience. But up to this point in our deep dive, we have only been discussing how to navigate a single static entity, the reference human genome. We've been exploring a generic, standardized map.
The composite average, yes.
But the true power of genetics lies in variation. The entire field of clinical genomics is driven by the need to understand how an individual patient differs from that standard map. What happens when our perfectly navigated location on the reference genome needs to be compared to the messy, mutated reality of a disease state?
And that takes us to the final, most complex section of the notes you provided, cross-referencing, utilizing VCF overlays and local alignment tools.
Right. Let's get into VCF files.
So when a clinical laboratory sequences a patient's DNA, they do not generate a brand new, massive 3 billion letter text file from scratch. The computational and storage overhead would be catastrophic.
You'd run out of hard drives.
Exactly. Instead, the sequencing software aligns the patient's data against the reference genome and only records the exact points where the patient's code diverges from the standard map.
It only records the typos.
Exactly. Every single divergent base is logged in a highly specific standardized file called a variant calling format, or a VCF file. This file acts as a catalog of mutations. It lists the SNP's single nucleotide polymorphisms, which are simple one-letter typos where a C is swapped for a T. It also lists the INDLs, insertions, and deletions where chunks of code have been erroneously added or completely deleted.
So how does a researcher actually visualize this in the Visual Genome browser? Because opening two separate browser windows and trying to stare at them side by side to spot a one-letter difference among thousands sounds like a total nightmare.
It would be impossible. The software solves this through data integration. Instead of opening a new window, you load the compressed VCF file as a direct visual overlay on top of your currently navigated view. The standard reference genome remains the structural foundation, the map underneath, but the patient's variant data is draped over it like a transparent sheet.
The source material specifically highlights an example using an individual from the 1000 Genomes Project, a massive public dataset, individual NA12878.
Yes, a very famous, heavily studied genome. When you load NA12878's VCF file while you are zoomed in on a specific gene, the visual interface completely shifts its focus. The underlying reference DNA is still visible for context, but prominent visual markers populate the screen, explicitly highlighting every single variant present in that specific individual's copy of the gene.
But wait, humans are diploid organisms. We have two copies of every single chromosome, one inherited from our mother and one inherited from our father. So a patient doesn't just have one single variant for a gene, they have two distinct alleles.
And navigating those dual alleles is where the software's contextual user interface design really shines. Recall the less than and greater than keyboard shortcuts we just discussed for jumping between exons.
Right, the DVD chapter skip.
When you have a VCF patient overlay active and you position your mouse cursor over the high-resolution DNA view, the software dynamically reassigns the function of those exact same keys. Pressing the less than or greater than keys no longer chapter skips between exons. Instead, the keystroke instantly toggles the visual display between the maternal allele and the paternal allele at that exact coordinate.
Now, I have to stop and push back on this design choice for a second. Let me make sure I understand the workflow. I've used my predictive search to find my target gene. I've used my display filters to cut out the pseudogenes and the noise. I've loaded my patient's massive VCF file. I'm actively analyzing the data.
Okay, I'm with you.
If the less than and greater than keys now automatically switch between the mom and dad versions of the gene, how do I go back to jumping between the exons? Do I just completely lose my structural navigation tools the minute I load a patient file?
It's a very perceptive question, and it addresses a core philosophy in bioinformatics interface design: context dependency. You absolutely do not lose the ability to navigate exons. The function of the keys is dictated purely by your active menu selection and cursor position.
Oh, so the software watches where my mouse is.
Precisely. If your cursor is hovering directly over the DNA sequence panel while the variant overlay is active, the software makes the logical assumption that your primary immediate focus is comparing the two alleles.
Makes sense.
But if you move your mouse down to the protein view panel, or if you right-click and explicitly select the exon navigation context menu, the software instantly reverts those keys back to jumping through the gene's physical structure.
That is brilliant. It intuitively maps the physical keyboard to the most biologically relevant structure based entirely on what you are actively trying to analyze in that specific moment. If you are focusing on patient variants, you are toggling alleles. If you are focusing on RNA splicing, you are toggling exons. It completely minimizes the need to endlessly click through drop-down menus. Your hands never have to leave the keyboard while you are navigating the biology.
And the visual feedback provided for these variants goes far beyond simply highlighting a changed letter. If a variant happens to fall inside a crucial coding exon, the software automatically translates the implication of that change.
Because changing a DNA letter often changes the amino acid that gets built into the final protein.
Exactly. In the software's protein view panel, any amino acid changes caused by the patient's variant are aggressively highlighted with bright magenta rectangles. The software immediately flags the physical consequence of the genetic typo, but it goes deeper.
How so?
If you hover your mouse cursor over that magenta box, a tooltip dynamically pops up. It tells you exactly what the amino acid change is, but more importantly, It utilizes an internal algorithm to predict if the mutation is a conservative or non-conservative missense mutation.
Can we explain the chemistry of that? Because I think that's the difference between a harmless mutation and a devastating disease. What is the software actually calculating when it labels something conservative or non-conservative?
It is analyzing the chemical properties of the amino acids involved. Amino acids are the building blocks of proteins, and they come in different shapes, sizes, and electrical charges. Some are hydrophobic, meaning they repel water and tend to fold into the center of a protein. Some are hydrophilic and like water.
So a conservative missense mutation would be like swapping one Lego block for a different Lego block of the exact same size and shape.
Precisely. If a variant swaps a small hydrophobic amino acid for another small hydrophobic amino acid, the overall 3D folding structure of the resulting protein probably won't be severely impacted. The software flags this as a conservative change. But a non-conservative mutation is like trying to swap a standard square Lego block for a large round wheel
The
piece.
entire structure breaks.
Yes, if the variant replaces a small and negatively charged amino acid with a massive positively charged one, the physical structure of the protein will likely collapse or malfunction. The software cross-references the chemical properties of the swap and flags it as non-conservative, providing the researcher with immediate, predictive, functional context for a raw sequence variant.
It does the heavy lifting and chemical analysis for you. But the cross-referencing capabilities of this browser extend far beyond just looking at human patient data. The notes also detail a highly powerful alignment view that allows for cross-species navigation.
Right. Local sequence alignment is a cornerstone technique in comparative genomics. It allows researchers to visualize evolutionary divergence.
The source material provides a fascinating example, comparing a sequence of mitochondrial DNA from a horse to the corresponding sequence in an elephant.
To execute this, a researcher could copy a small segment of DNA, perhaps 80 base pairs from the horse genome, place it in the search box, and then change the active database from the horse to the elephant genome. They navigate to the homologous region in the elephant DNA, highlight it, and command the software to generate a local sequence alignment.
The software then generates a specific visual panel showing the two distinct sequences stacked directly on top of each other. It explicitly draws lines and color codes the interface to show you exactly which bases have remained perfectly conserved across millions of years of mammalian evolution and exactly which bases have mutated and mismatched. You are literally finding the evolutionary divergence point right there on your screen.
But the most intense, multifaceted example of cross-referencing in the notes involves viral genomes. The viral comparison workflow is a masterclass in combining every navigational tool we have discussed so far.
I want to walk through this step by step because it really brings everything together. Imagine you are a virologist and you are researching the Zika virus. You open the Visual Genome browser and load the complete Zika sequence.
Now, we need some biological context here. Zika belongs to a family called flaviviruses. The fascinating and somewhat terrifying thing about these viruses is how they manage their genetic code. They don't have separate, distinct genes with their own individual start and stop signals. Instead, the virus translates its entire genome into one single, massive, continuous protein chain called a polyprotein.
So instead of printing individual tickets, it prints one massive, unbroken roll of tickets.
Exactly. And once that massive polyprotein chain is created, the virus uses specific enzymes called proteases, which act like molecular scissors, to physically chop the long chain into smaller, functional individual subpeptides that go on to replicate the virus and cause disease.
The Visual Genome browser actually understands this unique biology. It can detect the boundaries where those protease scissors cut. The notes explain that you can use the clipboard tool specifically by holding the Control key and double-clicking on the sequence to cleanly extract the precise amino acid sequence for just one of these functional subpeptides from the massive chain.
Let's say you use that tool to extract the RNA-directed RNA polymerase.
Which is the crucial engine the virus uses to copy its own genetic material. So I now have the Zika replication engine copied to my clipboard. Next, I navigate entirely out of the Zika database and I loaded the genome for a completely different virus,
Boy.
yellow fever.
Yellow fever is another flavivirus, structurally similar but evolutionarily distinct. Once loaded, you open the advanced comparison menu and initialize the Find Alignments tool. You paste the Zika sequence from your clipboard. You are fundamentally asking the algorithm, take the specific replication engine from Zika, scan the entirety of the massive yellow fever polyprotein, and find the structural machinery that is the closest evolutionary match.
And the software processes the alignment and returns a list of statistical matches in a structured result tree.
And this is where the intuitive navigation system brings the entire workflow full circle. You examine the results tree and double-click the top statistical match. The software instantly snaps your high-resolution view directly to the coordinates in the yellow fever genome where that matching replication engine resides.
It teleports you to the homologous location in a different species.
Mhm.
But it doesn't just dump you there and leave you to figure it out. It simultaneously generates a visual dot plot. This is a graphical matrix that visually maps the alignment between the two viral sequences, and it heavily color codes the amino acid sequence on the screen.
Looking at that color-coded alignment, you can physically see the conservative substitutions, the places where evolution swapped one amino acid for a chemically similar one without breaking the viral engine contrasted against the non-conservative destructive mutations.
By utilizing these tools in concert, you have gone from staring blindly at a raw genetic map to executing a highly complex cross-species evolutionary alignment, and it was all driven by the exact same core navigational principles.
It's just incredibly powerful.
I want to take a moment to pull all of this together because the scope of what we have covered, based on your notes, is massive. We fear this deep dive staring at a completely incomprehensible wall of text. 3 billion letters of raw, unformatted code. It was the ultimate data haystack.
But by mastering a specific computational control panel, that haystack becomes manageable architecture. By strategically dropping color-coded spatial markers 1, 2, and 3, we created a geometry we could measure and navigate. By utilizing raw pattern searching with C2LOF and predictive index-based gene teleportation with C2LG, we gain the ability to move instantly across the vastness of the map.
We deployed display filters like minus and protein plus lati to slice through the visual clutter, muting the noise and forcing the software to only display the specific biological structures we cared about.
We leveraged motif wildcards, brackets, and mismatch allowances to hunt down ancient, degraded jumping genes, utilizing algorithms like Boyer-Moore to bypass the immense computational load. And we learned how to physically interact with the biology, toggling through the fractured exons of a gene, or comparing the maternal and paternal alleles of a patient's VCF file simply by using the less than and greater than keyboard shortcuts.
When you look at it holistically, a researcher can now navigate the impossibly complex landscape of the human genome with the exact same fluidity, speed, and spatial awareness that you use to navigate a modern, sprawling city on a smartphone app.
And that capability matters profoundly. Whether you are a virologist hunting for homologous protein engines between Zika and yellow fever, an oncologist trying to visualize a heat map of severe gene dysregulation in lung cancer tissue, or simply a deeply curious learner trying to understand the mathematical repetitive structures capping the end of chromosome 1. Well, effective navigation is the only barrier against complete information overload.
It is the mechanism that takes a paralyzing, useless sea of raw data and distills it into actual, usable, functional knowledge.
Without a robust system to navigate, the data holds no value. The map is only as powerful as your ability to traverse it.
Which brings me to a final thought for you, the listener who sparked this entire deep dive. Throughout this conversation, we have been talking almost exclusively about navigating the reference human genome, a generic, standardized map built from a composite average of anonymous people. But the technology is accelerating. As the cost of personal whole genome sequencing continues to plummet, we are rapidly approaching a future where you won't just be navigating a generic reference map.
The tools will be the same, but the terrain will be yours.
Exactly. I want you to imagine loading a ultra-compact .2-bit file of your own personal genome into a visual browser like this. Imagine applying these exact filters to yourself. When the map you are navigating is the literal physical code that built you, what biological motifs, what ancient parasitic jumping genes, what unique inherited variations will you search for first?